The outline for that is a little more background about the virus by itself, in terms of it's the Senecavirus A or Seneca-Valley virus, and then we are jumping on the Brazilian experience on that, and how we deal with that, how we investigate it in terms of diagnostics and differentials there, and then move on for some of the cases that we have seen here, and also some projects that we are working on that to figure that out exactly. Clock postulate or other basic questions that we're still working on that, or still struggling in get that answer for specifically for Seneca-Virus Associated disease. And after that, we will also touch a little bit about the comparison between some antigenic comparison and some genetic comparison or characterization for the virus that we found here. Really briefly here, we talk about Senecavirus A. And they are proposing a new name, I believe it was last year, and then they started calling it Senecavirus A, but the common name still remain according with the taxonomy of the viruses. So either way, SVV or Senecavirus A, we still understand what we are talking about, and for this presentation I'm going to refer as a SVV for this condition here that I presented that. So the first full characterization of the virus was really in 2002. Well, it doesn't mean that it was the first isolation. As Daniel mentioned, we have isolated a picorna-like particle since 1988. And we have some case here at the VDL that we have those type of isolation too, picorna virus-like particle. And at that time, we don't know exactly what virus we are dealing with. And in 2002, for a cell culture medium contaminated, and they are at the investigation on the project and the paper, they are not really sure where it comes from, and they are speculating as porcine trypsin or fetal bovine serum. And the virus by itself, it's smaller than the ones, at least PERS and PED that we are used to work with. Just to be a nice comparison here, it has about 7K, and PERS and PED is a lot larger than that, and the PCV2 on the other hand, it's more smaller than that. So just to give you a reference on what type of virus we are dealing with. It's a picornaviridae family virus, and that's similar to FMD and swine vesicular disease. It's encoded a polypeptide, big one. It's cutting by itself and make those proteins. One of the important ones that Daniel showed in one of the trees is the VP1, and we're going to focus on the VP1 for comparison and sequencing. We also have done some whole genome sequencing for that. And this is how it looks like at the EM, one of the cases that we have for neonatal loss here. And in terms of the distribution, Daniel already mentioned that I'm missing here one of the states in Brazil. It's has been associated with vesicular disease in other parts of the world, and in Brazil has associated with the neonatal loss and vesicular disease, also with the spreading, as Daniel mentioned, was fast. And the difference pretty much between these two maps here, and we are talking about the US on the orange states here, we are talking about detection of the virus, not necessarily disease. And I point out here a couple of them that's more associated with disease. From 2012, one case in Indiana associated with exhibiting pigs or showing pigs in a state fair type of pig, showing vesicular lesions, showing coronary band lesions, and then the other states here associated with either neonatal loss or either vesicular disease. As Daniel already mentioned too, there has been a sporadic case since 1988, according to the NVSL, around 30 cases, including positive, some isolations. So we are here at this point, we are not talking about a brand new FAD disease, FAD investigation. The virus has been around. The big question is why it has been around for so long, and now we're starting not more seeing those sporadic cases and seeing more type of outbreak type of case. Here, closing that on the background, and here is more our experience in Brazil in terms of diagnostic investigation. This is more the first case that I was aware there. We received the samples on October 12 with complaining of acute neonatal mortality, of feed souse but no fever. And then on November 4th, which if you do, Daniel talk about the course is really fast and transient. If you do the math, it will be more than a week, but here maybe it's just November 4th because was the vet visiting that. So he knows that the huge difference. So it's a acute disease that goes away pretty fast. That's the main message that we got from there. And then we did some, of course, diagnostic investigation and all the things that we can think. And then we explored the diarrhea was the first complaint for the vet, and then you go for the reproductive and respiratory, and comes up nothing there. And then the only thing that we really found was rota B and C and Clostridium perfringens A. And when we went back to the vet and talked about our findings, that they looking us as a diagnostician out there and said the acute mortality, and they look at that and come on, you are better than that. Clostridium perfringens A is that huge mortality. And that's pretty much all I got. And it keeps going with that. And on the other case, sometimes we got some secondary change, and sometimes we got myocarditis, and we got a little more excited, and then we got PCV2 positive. As we know in neonatal piglets, it could cause myocarditis. Some ASUs, and Daniel mentioned about the colitis for difficile. It is not consistent. In some cases we have, in some cases we don't. Rota A, B, and C also associated, but not always. So that's the type of scenario that we kind of saw there, and we're still seeing here in case here in the US too. So, and the message, maybe the take home message here, if you're not looking around the whole or doing a good or complete workup for diagnostic, you will associate the mortality with Seneca and all the time. And as Daniel mentioned, some of the pigs may be healthy and carry or be positive for Seneca. So let's make sure that we have a really good or a complete workup in terms of diagnostics. And then we found nothing at that time, and then we started looking and speculating non-infectious toxicity, residuals, feed, mycotoxin, and then I started getting report about lameness in sows along with the neonatal loss. Daniel talked about that too. And then pretty much the red flag ups, and then we started thinking more about reportable disease investigation. Then the workup for that, including the 4 major diseases that we already discussed here, was done there, and nothing was found. And then 4 major diseases rule out, and then we jump on to looking for what we call, or the guys on the PCR lab you like to call unknown pathogen discovery through the next generation sequencing. And then we jump in on that and sequencing pretty much everything that we got in the sample. And then before that we retest for the 4 again and rule out. And then we come up with things like that, and I'm gonna take a couple minutes here because I think this is one of the tools that we are kind of have to get familiar with, the next generation sequencing, the sequencing, and how to interpret that. We have done that with PED and some rota, and Doug Martler is the main head in terms of interpreting data, in terms of working with this type of sequencing. And just to give you an idea in terms of our PED and rotavirus experience, when we got a CT of lower than 10 or around 10 or around 15 for rota or for PED, the amount of sequencing that we got is usually 1% or less than 1%. Which means that most of the sequencing that we have, it's low quality, and most of them, which is kind of expected for the host. So pig RNA, pig DNA surround here, bacteria, so we are taking out pieces on the pizza here, and then we work with just a small piece, which is SVV type of sequencing or matching sequencing to SVV. And then we end up found this 18% and 11% in the serum and the vesicular fluid, which is kind of surprised us in terms of the amount of sequencing of SVV in those samples. So we are more excited about the viremia, as team asked for Daniel, that we found about, I would imagine it would be in the peak of the clinical signs, 11% here a lot of sequencing, so we are more confident in terms of that association between Senecavirus and the vesicular disease. So just characterize the PCR and the distribution of the virus within the tissues. In terms of vesicular disease associated with Seneca, we found virus and the lowers will be, of course, on the lesions. So we may found here a CT of 12, 10, and then throughout all the tissues we can find some of the virus too, including brain. So serum, some viremia going on. And in terms of the neonatal piglets or the neonatal mortality, we also may be able to find virus throughout the whole tissues, and some of the CT is really low on the spleen, liver, and then we are working here more with the tissue homogeneity. Time. And so we have that picture in terms of virus distribution and the virus finding in terms of the PCR and the clinical signs, and when we want to move forward in terms of how is associated the virus within the lesions. And moving now with the experience here in the US and based on the experience that we have in Brazil, we work with Paul Soderberg in the Swine Health Information Center, and we put out some data for the screening PCR for oral fluids from about a little more than 400 cases, and we found 5 positive cases. And that was interesting because if you think more about the Senecavirus has been around for years, and we have experienced, for example, PCV2 a few years ago, where we have the most of the herd exposed to PCV2, it was kind of expecting that maybe the majority of the animals will be positive, just like we have a type of PCV2 type of picture, but it was not like that. It was really just a small piece of it. There was 5 cases in 5 states that was found positive for oral fluid. So, and I'm sure if you run oral fluid for PCV2 here, you're going to find a lot more. So that's not the case in terms of distribution for Seneca, and it was kind of a surprise to me at least. In one of the cases, and Daniel already mentioned that this was a screening case, it was not a disease-associated case. So we are not talking about a case or disease-associated case, and then in one of the reports, went back there, the vet went back there and found some vesicular lesions. And this point out one of the main keys that I think are lessons learned or being learned is still or the case in Brazil and here in the US, in terms of continuing education, in terms of really training to look at lesions, to look at vesiculars, in terms of not missing anything. That first case that I mentioned, probably the guys down there missed some of the vesicular lesions on the sows, and we also have some examples here that in the US we also have missing some of the lesions on the sows when we started doing workup for the neonatal loss. So that's one of the pieces of the information important in terms of lessons learned, in terms of training and training and retraining to really look at for identification of the lesions and how submitted samples and how, of course, FAD is a priority in that case, and how the process to collection. And I think we may be important to move forward with that. Here's the states that we're looking in terms of the screening and the positive states. That doesn't change much from the previous historical picture or map. In terms of kind of the same time we are doing PCR screening here, not using just oral fluids or our lab, we're using different type of samples, and it was randomly selected from the last year, pretty much based on the FMD surveillance and the CSF surveillance that we do in tonsils here. Try to get a snapshot, also kind of the same objective for the oral fluids, to see what type of positive or type of distribution we got in terms of positive PCR. So we got we're sampling over 4,000 of samples and 4 positive cases with no information about vesicular disease, and missing a piece here. So, and one case was vesicular disease, rinsate case that I'm going to mention later on in the presentation, and 5 cases are neonatal loss, and all 5 cases I can't remember if really has any vesicular feature about that. Because if it was, we would take another direction in terms of sample collection and processing for FAD investigation. So again, another example to maybe missing some vesicular disease in this neonatal loss. So more information about vesicular disease, and one interesting, we got 2 cases and over 10 years ago that was isolated on the coronavirus particle, and we tested now for PCR and turned out to be both of case Senecavirus positive and strong positive. So we have a virus isolation here, and we have neonatal loss with no information about vesicular disease in South tube. This is one of the recent cases that we are working on, and we have vesicular disease in this site here at some point 3 weeks ago or so. And then one of the difference here in terms of our experience that we had in Brazil was more, Daniel mentioned about that, about being milder or mild lesions, and the prevalence and the incidence is not that high. But for me it was impressive here, in that case here, that the incidence was really high. And I'm going to show you a video here that Dr. Laura shared with me, and I found pretty interesting video here. So she's passing here in the crates, and I hope you guys can appreciate some of the really big lesion. But the summary in general, I believe the video has, I don't know, about 20 or so cells, and 18 or 17 has some of the lesions, and she stopped that at some point to show the best ones. Some of them didn't get, some of them got some milder or some, depending on the stage of the clinical picture. Here you got a big one, and this one was really impressive for me. It was a huge, or one or coalescent vesicles there, a little more. So it was one of the difference that we still have to understand more is that why some of the incidents was really mild and the other ones was really striking, like these ones. And we are following up in terms of sampling all of the sites here. So basically we have a guild source here that goes here, guilds goes here, and then this F1 and F2 commingling nursery, and we need to finish in barns. So those pigs here, and they select some of the pigs here to go back here, so they have your own replacement. So they're going to be interesting to see how is the dynamic of the infections here. So we are doing oral fluid, serum, fecal swabs. Guys are working hard there to keep them up. In terms of the question that Daniel mentioned was detection versus disease. And then he mentioned about the cock postulate not being fulfilled, and the detection of the SVV within the lesions has not been fully shown. So we have a characterization of the virus in terms of isolation. We got this antibody from the Ming Yang from Canada, which is a hybridome supernatant with low titer. It's a good tool for characterizing the virus in culture after you isolate that. But for identification of the virus in tissues in terms of IHC, because of the low titer, we have some difficulties on that. And for that reason, we're jumping out for the in situ hybridization, which is pretty much has the sensitivity of the PCR, is a primary-based approach, and the probe hybridize the genome of the virus, and we can see that on the slides. Okay? So we are now trying to answer the questions in terms of causality, SVV detection here and disease, SVV detection here and disease. And we started for the easier one, which is here, which has been more strong association with that. This is a pilot study with IEC. We got a lot of non-specific binding here. Everything that you saw here, homogeneous and amorphous, it's the type of non-specific staining that we moved. That's one of the reasons that we're moving on. And I put this picture here. Terry has a picture, Daniel has, and I have the picture. So it's become famous, that one. So So the in situ hybridization is much more clean than the IHC, and we may appreciate here. This is a vesicle, a mature vesicle as this one, like here, and we got some microvesicle, I would say, like epidermal vesicles, like here, right under the epidermis, and there are some staining in the red here. Everything is red, is virus replicating that. And it's similar to FMD when we got a stratospinosum type of tropism. And here is the virus on the lesions that we are really happy with the ability of that technique to detect the virus within the lesion. So we are comfortable to say an associated disease with the presence of virus and within the lesions in that case. The puzzle here is the neonatal loss. So we have positive PCRs and we have the complete workup in terms of diagnosis, and we try to stain it with the virus. And the virus, actually there's a lot of virus there, but no lesions. And then we start to speculate in acute viremia and how the pigs are dying. And you walking through the tissues, we have small point, I don't know if you guys may appreciate that on the back, but there's a small diffuse presence of the hybridization of the virus on the liver. And we got this necrosis, or foci of necrosis in the liver. By the way, this liver was positive for A. suis. And us as a pathologist, we are looking for the virus on the necrosis, and there's no virus there. That was kind of disappointing for us. So the virus it has been difficult to associate with the lesions at this point. So more virus on the heart, some of the virus on the jejunum, and we have the diarrhea type of clinical signs, but no lesions, just the virus there. Maybe you want the entry for the virus. Still study on that. We compare the antigenic for IFA, so we have a monoclonal antibody from that group in Canada, and we have a reaction for all the isolates that we tested, the historical ones, the Brazilian and the US new ones. So we have some similarity in terms of cross-reaction here. This is the IFA. And this is maybe the most dangerous slide that I'm putting up here, because I'm putting up here that we are doing serology, and in fact, and the most important word here is still in development, and I'm afraid to put it here, and tomorrow I'm going to get thousands of serums to test that. That's not the case, okay? We are selecting the best cell line, and we establish the best titer for dilution, and we are progressing on there, some promising results, as you can see on the pictures. So I'm finishing up with the genetic comparison here. Daniels mentions about the VP1. Here's the whole genome, and the partial sequencing, that's the one that we have available. The blue one is the one that has an NVS cell, and we are working on that to try and get some funding to do the whole genome sequencing. The red one is the one that we do have whole genome sequencing, and the orange one is the one that we are working for that. So Daniel mentioned about that. We have pretty much two branches of two lineages here for the virus. And this is the old one, but old type of lineage. But we have some old here, 299. So we have a different type of evolution, I would say, in that reaction. In terms of comparison, we have among the Brazilian isolates, we have 99.5% similarity for the whole genome, and we have 99% among the US new or recent case. And then we compare both, and we have 98% of similarity. Is that too different? Is that same? I don't know. Actually, we have so few data out there, and we need to kind of build that database to trying to figure out what's the difference, what's the cutoff, we're working on that. And what we know is that we're talking about 98% of the whole genome, which is more or less 140 nucleotides. That a little different in terms of if you think about other viruses, and if you think about amino acid sequencing and the risk to have a different type of amino acid sequencing, a different type of protein encoding in that region. So that we're still working on that, but that's so far what we got. And then we move on forward for the 2011 isolates, and we have 96% similarity, and 2001 isolate, 94% of similarity, talking about the whole genome. And the VP1 just changed. If you look at these guys here, they're going to go to the bottom now. But the pattern is the same. But what is interesting for us is the comparison pretty much the same. We got 97 between the US and the Brazilian, and 29 nucleotide. Is that a lot? Is that few? We're still struggling to answer that question yet. And 95 and 96% with the 2011 isolates, and 94% with the 2001 reference isolate, or the California isolate, which is the feral, one of the feral pigs type of isolate. Okay, closing here. I probably run out of time. Lessons learned, or lessons being learned. Actually, that has been a great learning process for me personally. FAD investigation is still a priority. The economic impact for that so far that we have discussed is usually because of the FAD investigation in terms of closing packing plants, and we do have here a good logistics for diagnostics, and send them up to Plum Island, and then they can get the result right away, which we didn't have at that time, and I don't believe we have yet in Brazil. So the industry in Brazil is suffering a little more about this than us here in terms of diagnostics or response, rapid response. Continuing education, I mentioned before about investigation and training practitioners for investigation of vesicular lesions. Strong association, vesicular lesion, SVV, we're happy with that, but we're struggle to answer the question in terms of virus and no lesions. Short viremia, that we have a study of following up that it seems to be a short live viremia. Sequence interpretation, I have pretty much more questions than answers to you guys, so don't ask about that. And lack of information on basic question. And the other question, the incubation, shedding, transmission, and potential source, we're still working on that. So much more a learning process than really a final statement here. And the last but not least, we started reporting last week the case, or the new case, on the swine health monitoring project led by Bob Morrison. And we started last week, and then we are moving forward with that. That's one step ahead that we are envisioned for that specific syndromes and disease.